Slide 1. This slide tape is the fourth in a series of presentations on acid-base physiology, prepared by the American Physiological Society. 2. This program begins with a review of prerequisite knowledge. Before you go any further, make sure you have completed the entry skills test provided in the study guide. 3. During the presentation, question slides will appear at intervals. Stop the tape to answer the questions, then advance to the next slide on which the correct answer will appear. Stop the tape again to check your answer. After you finish the unit, assess your understanding of the material by taking the post-test in the study guide. 4. The subject of this discussion is renal regulation of hydrogen ion metabolism. Content expert, Dr. Jordan Cohen. 5. This unit will discuss, first, the source of hydrogen ions in body fluids. Second, bicarbonate debt. Third, mechanisms by which bicarbonate is conserved and generated. Fourth, the formation and excretion of acid in the urine. And fifth, a review of factors which affect bicarbonate concentration. 6. In the unit defining the role of respiration in hydrogen ion metabolism, you learned that PCO2 and the concentration of carbonic acid are controlled by the respiratory system through its regulation of carbon dioxide excretion. 7. In this presentation, you will learn that the concentration of bicarbonate ions is controlled by the kidney through its regulation of hydrogen ion excretion. 8. The keystone in the regulation of extracellular hydrogen ion concentration is the carbonic acid bicarbonate buffer system. The significance of the carbonic acid bicarbonate system stems from its being the most prevalent of the buffers present, and even more importantly, from its being the only buffer system in which the concentration of the components may be varied independently by physiological regulatory mechanisms. 9. The ability of the body to adjust these variables independently permits this buffer to function as the crucial link between the purely chemical process of buffering and the exclusively physiological processes of acidity regulation. 10. The set of physiological processes responsible for adjusting the rate of hydrogen ion output in response to change in hydrogen ion input in order to maintain steady state conditions is referred to as the hydrogen ion regulatory system. 11. The control system regulating hydrogen ion balance differs from those regulating the balance of most substances due to an enormous disparity between the large amount of hydrogen ion that must pass through the body relative to the vanishingly low concentration of free hydrogen ions that may be tolerated by the living organism. 12. The first objective is to enable you to 1. Identify the neutral precursors of endogenous acids. 2. State the processes by which neutral precursors are converted to endogenous acids and name the products. 13. What determines hydrogen ion input? Hydrogen ions are normally introduced into body fluids by the process of endogenous acid production. By this process, neutral precursor substances are broken down by cellular metabolism to a variety of strong acids. In contrast to sodium and other electrolytes that gain access to body fluids directly as a result of ingestion, preformed acids are only rarely present in the diet. 14. Endogenous acids may be derived either from the breakdown of common constituents of the diet or from body tissues. Consequently, factors that alter the rate of metabolism may lead to variation in hydrogen ion input despite a constant diet. 15. The first and quantitatively the most important process contributing to the endogenous acid is the oxidation of sulfhydryl groups of cysteine and methionine to form sulfuric acid. Sulfuric acid contributes the major portion of the strong acids introduced into the body fluids by cellular metabolism. 16. The second process is the hydrolysis of various phosphoesters to form phosphoric acid. 17. The third process is the incomplete breakdown of certain fats, carbohydrates, and proteins to form organic acids, which may either be true end products of metabolism, such as uric acid and creatinine, or which may be metabolic intermediates, such as beta-hydroxybutyric acid and lactic acid. Under some circumstances, intermediates are generated faster than they can be converted to carbon dioxide and water. 18. The contribution made by the diet cannot be predicted precisely, and the contribution of catabolic processes cannot be measured directly. Therefore, it is exceedingly difficult to determine the rate of hydrogen ion input to body fluids with the same precision as is possible for the input of sodium and other electrolytes. 19. Nevertheless, it can be shown that a typical high-protein diet yields approximately 1 milliequivalent of hydrogen ion per kilogram of body weight per day, or some 50 to 100 milliequivalents per day for the average adult. 20. The kidney is responsible for adjusting the rate of hydrogen ion output from the body in response to changes in input, and thereby for restoring body fluid composition to normal. 21. Remember to stop the tape to answer questions and to check your answers. 22. 23. 24. 25. Our next objective is to be able to 1. State the mechanism by which bicarbonate ions permit a large hydrogen ion load to be handled by the body with a negligible change in hydrogen ion concentration. 2. Define bicarbonate debt. 26. Titration of body buffers permits the influx of a large quantity of strong acid, such as that produced each day by cellular metabolism, to occur without a significant change in body fluid acidity. The addition of this quantity of acid to a solution lacking in buffers would result in a large increment in hydrogen ion concentration. 27. Note that buffering itself makes no direct contribution to external hydrogen ion balance, since this process does not remove hydrogen ions from the body. The critical role of buffers is confined to blunting the impact of endogenous acid production on hydrogen ion concentration. 28. Among the immediately available buffers, bicarbonate is the most prevalent and accounts for about 50% of the hydrogen ions that gain access to body fluids. During the titration of body buffers by hydrogen ions, bicarbonate is converted to carbonic acid, and then to carbon dioxide and water. As a consequence, the number of bicarbonate ions in the body is reduced. The carbon dioxide generated by this titration is added to the very much larger quantity derived from cellular metabolism and is excreted by the lungs. 29. The remaining hydrogen ions combine with other hydrogen ion acceptors. The participation of other body buffers is governed by the isohydric principle. As a result, not only are bicarbonate stores depleted, but some non-bicarbonate buffer bases, such as plasma proteins and hemoglobin, are converted to their respective acid forms. 30. Bicarbonate debt is defined as the quantity of bicarbonate ions required to fully replenish body alkali stores. Because the process of titration is reversible, this quantity is equivalent to the amount of hydrogen ions originally added, regardless of the relative participation of the various buffer systems. 31. As if you hadn't guessed by now, the kidney is the source of the new bicarbonate ions used to repay the bicarbonate debt, and thereby makes its crucial contribution to hydrogen ion homeostasis. The mechanisms responsible for this regulatory function are described in detail in the next section. 32. 33. 34. Your third objective is to 1. Complete a diagram which specifies the mechanism by which the kidney both conserves existing bicarbonate and generates new bicarbonate for the body fluids. 2. Give percentages of bicarbonate reabsorption along the nephron and the rates of sodium-hydrogen exchange required to 1. Prevent excretion of filtered bicarbonate, and 2. Generate new bicarbonate. 35. Before answering the question of how the kidney repays the bicarbonate debt incurred during the process of body buffering, it is necessary to consider the kidney's critical role in conserving the bicarbonate ions presented to it in the glomerular filtrate. 36. Bicarbonate and sodium ions are freely filtered at the glomerulus. The rate of bicarbonate filtration is given by the product of the glomerular filtration rate and the bicarbonate ion concentration in plasma water. 37. The sodium ions filtered with bicarbonate are returned to the body fluids directly by reabsorption across the tubular epithelium. 38. Filtered bicarbonate ions are also returned to the body fluids. However, bicarbonate cannot be reabsorbed directly because the luminal cell membrane is impermeable to bicarbonate. This constraint is neatly sidestepped by the presence of a very effective mechanism of bicarbonate conservation. 39. Conservation of filtered bicarbonate is initiated by a process referred to as sodium-hydrogen exchange. When those sodium ions that are associated with bicarbonate in the glomerular filtrate are reabsorbed from the luminal fluid by the tubular cell, electroneutrality is maintained by the secretion of an equivalent amount of hydrogen ions into the tubular filtrate. 40. When the filtered bicarbonate ions that are left behind in the luminal fluid encounter these secreted hydrogen ions, they are converted to carbonic acid. The carbonic acid thus formed quickly decomposes, yielding carbon dioxide and water, which are readily recycled. 41. The secreted hydrogen ions are made available by the dissociation of cellular carbonic acid derived from the hydration of carbon dioxide. The rate of this reaction is accelerated by the catalytic influence of the enzyme carbonic anhydrase, which is abundant in the tubular cells of the kidney. 42. Finally, for each hydrogen ion secreted by the tubule cell, note that a bicarbonate ion is also generated. These newly formed bicarbonate ions, rather than those originally filtered, are the ones conserved by the kidney and balance the sodium ions reabsorbed via hydrogen ion exchange as they pass into the peritubular fluid. Therefore, the overall rate at which sodium ions are exchanged for hydrogen ions is equivalent to the rate at which the kidney returns bicarbonate to the body fluids. 43. Each of the nephron segments concerned with bicarbonate conservation appears to utilize this indirect mechanism of bicarbonate reabsorption, but differs markedly in its quantitative contribution to the overall process. The proximal convoluted tubule is the most important site of hydrogen ion exchange and bicarbonate conservation. Clearance studies and micropuncture experiments indicate that the bicarbonate ions conserved by the proximal tubule account for 90% or more of the filtered bicarbonate. This observation correlates well with the abundant carbonic anhydrase activity that can be demonstrated at the luminal surface of proximal epithelial cells. 44. The loop of Henle appears to play a minor role in bicarbonate conservation. Therefore, if bicarbonate excretion is to be prevented, the distal convoluted tubule and collecting ducts must remove whatever bicarbonate remains in the luminal fluid. 45. If the rate of sodium-hydrogen exchange were equal to the rate of bicarbonate filtration, the available bicarbonate ions would merely cycle through the kidney without increasing their number in the body. If the kidney did nothing more than prevent the excretion of filtered bicarbonate into the urine, no net contribution to body bicarbonate stores would result. 46. To repay the bicarbonate debt, additional bicarbonate is required. The kidney must return more bicarbonate ions to the body than it receives in the glomerular filtrate, thus requiring that the rate of sodium-hydrogen exchange exceed the rate of bicarbonate filtration. 47. Let us consider the actual rate of sodium-hydrogen exchange required to conserve the bicarbonate ion arriving in the filtrate. At a normal filtration rate of 180 liters per day and a normal plasma bicarbonate concentration of 25 milliequivalents per liter, approximately 4,500 milliequivalents of bicarbonate are filtered per day. Therefore, the rate of sodium-hydrogen exchange required to prevent excretion of the filtered bicarbonate is approximately 4,500 milliequivalents per day. 48. Contrast this very large baseline of sodium-hydrogen exchange with the relatively small amount of new bicarbonate ion that the kidney must generate to repay the bicarbonate debt created by endogenous acid production of 50 to 100 milliequivalents per day. 49. 50. 51. 52. 53. Our fourth objective is to be able to 1. Identify the processes by which hydrogen ions are excreted when they can no longer be recycled through carbon dioxide and water, and define the role of urinary acidification in these processes. And 2. Define net acid excretion. 54. Once sufficient hydrogen ions have been secreted by the kidney to prevent excretion of filtered bicarbonate, the secretion of additional hydrogen ions corresponds to the amount of new bicarbonate that is generated. These hydrogen ions cannot be recycled through carbon dioxide and water because bicarbonate ions are no longer present in the filtrate. They must be accounted for in the final urine in another way. 55. A small but physiologically critical fraction of these hydrogen ions remain in solution and increase the urinary hydrogen ion concentration. In man, maximal urinary hydrogen ion concentration is approximately 0.04 millimoles per liter, which corresponds to a urinary pH of 4.4. Even though this represents a 1,000-fold increase in hydrogen ion concentration over that in plasma, only a trivial amount of hydrogen ion can escape into the urine as such. 56. As a result of the kidney's ability to generate an acid urine, secreted hydrogen ion is bound by filtered buffers and by ammonia, and is excreted into the final urine as titratable acid and ammonium ion. Taken together, titratable acid and ammonium excretion represent urinary hydrogen ion output. 57. The term titratable acid refers to the amount of hydrogen ion present in the urine in combination with the base components of all filtered buffer substances that are destined for excretion. 58. The rate at which urinary buffers convey secreted hydrogen ions into the urine is an interdependent function of 1. The nature of the various buffers present, and 2. The prevailing degree of urinary acidification. Normally, about 1/2 of the total amount of hydrogen ion excreted is in the form of titratable acid. 59. Phosphate is the urinary buffer that normally accounts for the bulk of titratable acid. At normal hydrogen ion concentrations in plasma filtrate, about 80% of this buffer solute is in the dibasic form. At the hydrogen ion concentration of maximally acid urine, however, virtually all of the phosphate excreted is in the monobasic form. 60. Ammonium is the other major repository of hydrogen ion in the urine and has even greater physiological significance than titratable acid, since it responds more to alterations in hydrogen ion input. Urinary ammonium ion has its origin in ammonia generated within renal tubular cells, largely by the deamination of glutamine. This process is catalyzed by the enzyme glutaminase. 61. Two important properties of ammonia contribute to its role in hydrogen ion excretion. First, ammonia is an unionized substance and hence diffuses freely across cell membranes. Second, ammonia has a very great affinity for hydrogen ions, that is, it is a very strong base. As a result, ammonia removes secreted hydrogen ions from solution with great efficiency. In so doing, freely diffusible ammonia is converted into non-diffusible ammonium ion, which cannot move back across the luminal cell membrane and is thereby trapped in the urine. 62. As hydrogen ion concentration rises in the tubular filtrate, the concentration of free ammonia base in the urine must fall, enhancing the net rate of transfer of ammonia into the urine. Therefore, both the rate at which the renal epithelial cells generate ammonia and the extent to which the urine is acidified critically influence the degree to which urinary ammonium serves to convey hydrogen ion into the final urine. 63. So far, we have considered only those circumstances in which the body is confronted with the net addition of hydrogen ions, creating a bicarbonate debt, and hence the need to excrete hydrogen ions by the kidney. 64. Now consider the circumstance in which the body is confronted with a net addition of alkali, resulting in a bicarbonate surplus, and hence the need to excrete bicarbonate ions by the kidney in order to restore body fluid composition to normal. 65. To excrete surplus bicarbonate ions, the rate of sodium-hydrogen exchange in the kidney falls short of the rate of bicarbonate filtration, and thus permits some filtered bicarbonate to be excreted into the urine. 66. Therefore, the kidney can produce urine containing either excess hydrogen ions or excess bicarbonate ions. The concept of net acid excretion is useful in describing the overall contribution of the kidney to hydrogen homeostasis. Net acid excretion can be defined as the sum of ammonium and titratable acid excretion minus urinary bicarbonate excretion. Note that net acid excretion has a positive value when hydrogen ion output is preponderant and a negative value when bicarbonate output is preponderant. 67. 68. 69. Our last objective is to review those factors that can affect the bicarbonate concentration of body fluids. 70. The conversion of neutral foodstuffs by cellular metabolism, or the catabolism of body tissues, delivers hydrogen ion into body fluids, creating a bicarbonate debt. Ingestion of alkali, on the other hand, creates a bicarbonate surplus. 71. When the body is burdened by a net addition of acid and thus incurs a bicarbonate debt, the kidney restores body composition by maintaining a rate of sodium-hydrogen exchange in excess of the rate of bicarbonate filtration, thereby excreting the acid load into the urine. When the body is burdened by the net addition of alkali and thus experiences a bicarbonate surplus, the kidney restores body composition by excreting the excess alkali, resulting in a negative rate of net acid excretion. 72. Although of negligible concern to this system during health, it should be remembered that the gastrointestinal tract possesses enormous potential for disrupting hydrogen ion balance during certain illnesses. Loss of gastrointestinal secretions containing either hydrogen ions from the stomach or bicarbonate ions from the pancreas and small intestine can alter bicarbonate stores significantly and either counterbalance or compound the bicarbonate debt. 73. From this summary, we can now pinpoint the major factors that impinge on the bicarbonate concentration of the extracellular fluid compartment. These factors are: 1. The rate of acid or alkali input to body fluids. 2. The rate of net acid excretion by the kidney. And 3. The rate of hydrogen ion or bicarbonate ion loss from the gastrointestinal tract. 74. 75. 76. It is appropriate to conclude this discussion by refocusing on the physiological regulation of acidity. Plasma acidity is the byproduct of two physiological control systems, one regulating the partial pressure of carbon dioxide and the other regulating the concentration of bicarbonate ions. Hence, plasma acidity may be regarded as a dynamic physiological variable, fluctuating from moment to moment as the regulatory systems respond continuously to ever-changing demands. 77. This concludes the discussion of renal regulation of hydrogen ion metabolism. Refer to your study guide for the post-test and references for additional reading.